Thanks for having me here so that I can talk a bit about our more recent work on the evolution of cooperative virulence and how we have a mouse model to study aspects of this using Salmonella as a focal model. So we've talked a lot about different aspects of bacterial sociality already, so I'm not going to go into great amount of detail about this, but cooperation is one of these aspects. And I just want to point out here that cooperation can be found in many aspects of lifestyle for bacterial pathogens. And this can be through shared metabolites or enzymes in the context of biofilm formation, or as a consequence of virulence factors, which I'm going to be talking about in this talk. But in all these cases, what's important to note here is that the mechanism by which they cooperate is through a shared public good. And this is already a super old equation, but it still kind of fits this, these concepts that I'm going to be talking about. And this is kind of proposed to be the conditions that allow cooperation to evolve. And this is kind of summarized by the benefits to the receiver multiplied by the relatedness of the producer to the receiver. And this has to outweigh the cost of production. Okay, and because all of this functions through public good mechanisms and that there's a cost associated with this, it's not surprising that you can have mutants that emerge, and this can be interpreted as a decrease in relatedness, and they start to outgrow the producing strains because they no longer pay the cost of production. And this happens in all of these examples that I've shown you above. And again, to kind of reiterate this, so here we have depicted a producing cell that's shown in gray here with these little black dots, which are the public good. And then you have a white strain that emerges, which is in this case a cheater. And then these guys start to grow faster. But the problem with that is that all of a sudden you don't have enough public good around because there's less production. And the entire population can crash in some cases. So this is very clearly a problem and potentially a dead end. But as I mentioned, a lot of aspects of bacterial lifestyle, cooperation is found. So the question is, of course, how is cooperation evolutionarily maintained? And there are some theories that try to explain that. And one of them is mentioned in Jeff Smith's paper from 2001. And here I'd like to specifically thank Josh Thomas from yesterday's session to explain this in a very nice way. So thanks a lot for that. But let me just kind of summarize this again. And so the idea is that basically you can reinforce cooperation in a population of cheaters by actually conjugating the cooperative allele encoded on a mobile genetic element. And that's shown here by the entire population that starts cooperating again. And then one way to interpret this is by looking at increased relatedness in this in Hamilton's rule. Okay, and so he was saying that actually this can be relevant for cooperative virulence specifically, and that's because a lot of virulence factors are found on mobile genetic elements, and a lot of them have extracellular functions. Okay, so that we found very interesting, and then we thought we'd try to do this experimentally. So there have been some work in vitro and also theory to support different aspects of this hypothesis. But to really look at cooperative virulence, what's very important is to have relevant host infection models to capture the entire complexity of the system. So as Sarah introduced, in our lab, we work with Salmonella, and we have some good mouse models to look at aspects of Salmonella infection biology. And one of the things we know about Salmonella from this work is that Salmonella actually, once it grows up to a high enough density in the gut, it will actually invade into host cells dependent on its Salmonella pathogenicity island 1, also called SPY1. And when it does this, it leads to a host immune response, and this triggers inflammation, which acts as a public good to fuel growth of Salmonella in the gut lumen and suppress the growth of the microbiota. So it allows it to have a favorable niche. What we also know is that over time, you have cheater mutants that start emerging and overgrow the population, such that if enough of these emerge, you actually start losing the ability to trigger inflammation, and then the population of Salmonella starts to go down. And what we know is from previous work in our lab is that these cheater strains, the genetic locus for this is on Hill D, which makes sense because it's a central regulator for virulence associated with SPY1 in Salmonella. Okay. So putting these things together, I started to construct a system to actually be able to study the role of horizontal gene transfer in this. So I basically took a conjugative plasmid naturally found in one of our Salmonella strains, and I cloned in a functional copy of HIL-D, which is in this case the cooperative allele. And in the donor strain, I killed the function of the type 3 secretion system 1, which is very important because this is a secretion system where virulence factors are secreted through in this case. So if you don't have this secretion system, you can't engage in any of the virulence associated with SPY1. So this strain is essentially avirulent. And on the other side, I have a recipient strain, which is kind of the inverse scenario. There's no functional copy of HIL-D, but you do have a functional copy of the type 3 secretion system 1, such that if conjugation occurs, then you get a virulent strain, and only in this case. And so this is the mouse model we use to address this question. So basically, I antibiotic pre-treat the mice. 24 hours later, I add the donor and the recipient at low inoculum size to ensure that conjugation only happens in the gut lumen. I sample over time the fecal populations until the end of the experiment. And of course, the first question is the plasmid actually transferred? And what we can see here in red, the virulence plasmid, which I call PVIR, which encodes the functional HIL-D allele. And this is in contrast to control plasmid, which doesn't have a HIL-D allele. And you see that already after 48 hours, you start to see that almost the entire population has obtained the plasmid. And on the median, this is quite well maintained in the population. Okay, so it looks like horizontal gene transfer aspect works, but can it actually restore virulence? And for this, what we can do is actually monitor the state of inflammation in the mouse over time by taking a secreted host protein that is secreted in response to Salmonella insult. And when we do this over time, you see that inflammation starts to rise in strains where they do have a functional HIL-D allele on a mobile genetic element. So this is also striking because now we show that in this mouse, virulence can essentially be generated de novo because you have two initially avirulent strains that only when they're mixed together, they can trigger inflammation. But if you look at the entire time course of this experiment, you start to see that the inflammation goes down. So this is kind of a short-lived phenomenon. And so then we asked why this was the case. And in order to do that, we looked at the percentage of cells that can actually trigger SPY1. And so we have a pretty good readout for that. And with this, you start to see that at the beginning, you have a very high proportion of the population that can cooperate. And that's because this mobile genetic element, which was initially completely functional, got transferred to the population. But over time, you start to see that more and more of this population is not able to cooperate and that cheaters start to emerge again. And if you look at the last, if you take clones and you resequence them, you see that there's actually a mutation on the cooperative allele, so HIL-D on the mobile genetic element, which has implications for this reinforcement theory, because now all of a sudden you have a cooperative cheating, you have a cheating mobile genetic element competing with a cooperative one. So it's not actually helping that much in this case. Okay, so then we asked, okay, there's obviously other aspects of this equation. So one of them is cost. And so we asked, how much does the cost of virulence actually matter in this case? And so what I did is I basically cloned another variant of this PVIR plasmid, which I call the low cost variant. And so I just basically changed the upstream regulatory parts of HIL-D. And when you do this and you again perform the same experiment, you start to see that cheating is much slower. So if you look at the last day of the experiment on day 10 here, you see that you have much more cooperative clones when you use a low-cost plasmid compared to a high-cost plasmid. Okay, so if I summarize so far this, let's call it within host evolutionary dynamics, we have a wild-type population of cooperators which can be subject to cheating. This can be reinforced by a cooperative allele on a mobile genetic element. Leading to complemented cooperators, but they start cheating again, and this is dependent on the cost. So it seems like these aspects kind of only increase the time where you can have virulent clones in this case, or cooperative clones. But again, this is not the whole picture, so we of course have to consider what is the benefit of SPY1 in Salmonella. So I told you that it's basically to trigger inflammation, but the whole point of Salmonella to grow in the inflamed gut is to fuel their own transmission. And so what we need to look at is what is the influence of these cheating dynamics on transmission. So I basically performed the same experiment, but I took samples at day 2 and day 10 post-infection, and I did a fecal transmission into new antibiotic pre-treated mice, and then I monitored the progress as in the first mouse. And so what you see if you do this, if you transmit at a very early time point where you have a very high proportion of the population that is cooperating, you see that inflammation is triggered very nicely in both mice infected with the low-cost and the high-cost plasmid. And this is actually in contrast to when you do this on day 10, because on day 10, you have more virulent clones when in the low-cost plasmid. This leads to inflammation. And in the high-cost variant, you have a large amount of cheaters compared to cooperators, and this doesn't lead to disease. And this, of course, since I told you this has implications on shedding, if you look at the total population in the feces, you start to see that it actually starts to drop when you have a majority of cheaters in the population, in contrast to cooperators. But then if you look at the proportion of the population that again is cooperating in these new mice, you find a very large variability. And so it looks like cheaters start to emerge again and overtake the population. And this can occur either through new mutations or because you actually transmitted cheaters in the first place. And so if you think about it, transmission of the population doesn't really select for virulence. And that's because cheaters will always profit in the presence of enough cooperators. But then if you think of how transmission works in more realistic cases, I would bet that in most cases, you don't transmit an entire fecal pellet into an antibiotic pre-treated animal. So there would be arguably a larger host-to-host transmission population bottlenecks. And this comes from the environmental step in between transmission or cases where you have an intact microbiota and these clones have to actually fight to establish themselves. And so the question is, what can we actually artificially increase or make the transmission bottleneck more narrow? And what we can do is take evolved clones from the previous experiment and then infect them as single infections, kind of simulate this higher transmission bottleneck. And when we do this, and what I'm showing here is a pooled result of high cost and low cost clones, they don't really show a large difference, is that you see when you transmit cooperating clones, that's the SPY1 positive clones, you do get inflammation, and you don't when you transmit a cheating clone. And this again has implications on the shedding population. Okay, so if I want to kind of summarize in the context of Salmonella in our mouse model, sort of an evolutionary stable strategy for cooperative virulence, we have these two aspects of Hamilton's rule. So either increasing relatedness in this case by restoring by horizontal gene transfer, or decreasing costs. These are apparently only short-term solutions in this case. But if you look at the role of transmission, this is potentially long-term, and this can be dependent on transmission bottleneck. And if I want to summarize this a little bit more graphically, so you have a within host evolution mixture, let's call it, of cooperators and cheaters. And if you have a wide bottleneck, as is done in my experiments, this can allow for transient disease. And in this case, there's no real selection for the virulent genotype. But within more realistic cases with a narrow transmission bottleneck, you may select for the virulent genotype at the population level. And that's because when you have a cheating clone, this doesn't lead to any disease or shedding. And this is in contrast to when you transmit virulent clones. And with that, I'd like to thank everyone that's been involved in this project and helped me throughout my PhD. And I'd be happy to answer any questions.
Thanks, Erik. That's really an awesome data set. It's really cool to see how social evolution and cheating and cooperation can play out in a mouse. So there's one question that we have from the attendees. Hi, thanks for the great talk. Do you have some information about the number of generations in the growth cycle of a population? How many times are they dividing but in your different?
Yeah, so this is we have ways of measuring this. I haven't done it so far, but I can kind of estimate. It's a bit hard to estimate because the inflamed gut actually promotes both increased death and increased growth rate. So we know that they undergo generations quite quickly in this state, but in the non-inflamed gut, which happens kind of transiently, so it starts non-inflamed, goes to inflamed, and then it goes back to non-inflamed. So it's really hard to actually estimate how fast they're growing. But of course, in cases where they're more inflamed, they grow faster. So this can allow chances for mutation to occur more in this case than under the non-inflamed cases. So it may be that actually while fueling their own transmission, this actually speeds up cheating. So in the cases where it's beneficial.
So maybe I had another quick question. I know we're out of time, but I'm really interested in this. So you've shown that horizontal gene transfer can actually play an important role in maintaining cooperation, even though it's maybe a short-term solution. So do you think that your data supports or refutes what Josh was talking about yesterday? So this idea that cooperation can be maintained through horizontal gene transfer or not?
Yeah, I think it definitely plays a role. And this has to, again, going back to Josh's talk, it's a lot in the context of, let's say, bacterial lifestyle or the type of virulence factor that's encoded on these mobile genetic elements. Of course, what I'm using here is a super costly HIL-D gene, which is not normally found on a mobile genetic element, but there are many other virulence factors that can be found on mobile genetic elements and phages and plasmids, and there it may actually have a role. But it's very hard to measure that experimentally because the cost is so low that during the course of an experiment, you probably can't observe these dynamics.
I think the dynamics you observe are already quite clear. It's really nice.
Yeah.
Great. Well, thanks a lot for your talk. Thanks to all three speakers.